Welcome to the newest installment of the Methods in Enzymology webcast series. Today, you'll hear about the highly anticipated second edition of the Guide to Yeast Genetics volume, publishing in hardcover and paperback. In 2002, the original Guide to Yeast Genetics published, and it went on to be the top-selling MIE volume ever. Now, a completely revised edition of this classic volume is publishing under the editorial guidance of Jonathan Weissman and original editors Christine Guthrie and Gerald Fink.
The Guide to Yeast Genetics, as an example. A graduate student will come into a laboratory not having done yeast genetics, and almost all of the things that they need to do are covered in yeast genetics, and you will see that volume on their desk, and many articles in that volume are accessed by the student. The original Guide to Yeast Genetics was a great volume. It had all of the methods that one needed to do yeast genetics, and I've done yeast genetics. There are a lot of specific methods. One needs to know how to dissect tetrads. It's a difficult thing to do, and it's explained clearly how to do this. And so if you go to a yeast genetics lab, and I work in a yeast genetics lab, you will find that all of the students have a well-thumbed volume of the Guide to Yeast Genetics on their desk. Methods in yeast genetics have gone on, and in particular, big systems approaches. For example, of course, the entire genome of yeast has been sequenced, and that information can be farmed and accessed in a number of different ways, and it explains how to do that in this volume. All of the genes in yeast have been knocked out, that can be knocked out, how to use that library. All of the genes have been tagged with green fluorescent protein, making it possible to do all sorts of cytology on yeast, and so there are tremendous systems approaches. New approaches that use microarrays to look at, let's say, the expression of every gene in yeast or the splicing of every gene, pre-messenger RNA in yeast. All of these things are explained in this new volume. The editors continue to be Christine Guthrie and Jerry Fink, the editors of the first volume, but they've added Jonathan Weissman, a brilliant young yeast geneticist at UCSF, and it's Weissman who has pioneered many of these large array applications that are now so popular in the yeast field.
Many contributors have left their vast expertise to this eagerly awaited volume, and we sincerely thank them, along with the editors, Jonathan Weissman, Christine Guthrie, and Gerald Fink.
So full disclosure, I'm married to one of the, and it isn't either Fink or Weissman.
To learn more about this volume, read sample chapters, and view webcasts, visit us online at elsevierdirect.com/MIE.